because they might think that the barrier is unable to sustain the conflict or reduce the conflict basically. And in case of if compensation was provided to people in the areas with the barrier, then people were willing to pay further for the barrier. But in case of compensation for the non-barrier areas, then people were getting compensated, so they did not feel the need to invest in the barrier. Right. And with the increase in time taken for compensation, it was found that in the barrier areas, people were actually unwilling to pay due to their distrust in the functionality of the barrier, and complete opposite results were seen in case of crop breeding frequency and elephant incursion change in the barrier in the non-barrier segment. So this meant that people were actually they wanted barriers in their site so that the amount of or the amount of incidence of conflict can be reduced in their area. For management implications, in the area, I noticed that the solar fences were discontinuous in some parts and they were on non-uniform length across the entire stretch. And for trenches as well, the depth and the depth and the width of the trench varied in different parts of the entire stretch. And in some areas, the trench was also even filled up. This might be because of high siltation issue in the landscape of that district. And people also personally invested in low water solar fences and fences which were around 9 feet deep around their houses. And NGOs also provided provisions like makeshift machines and providing torches which were portable, rechargeable, and had a beam of which can go up to 2 kilometer range. And capacity building in terms of training for handloom and mushroom cultivation and vermicomposting were also provided to decrease dependence on agriculture. As a way forward, from my study, I can suggest that lemon cultivation along the fringed villages of the protected areas can be effective because generally elephants tend to avoid those areas. And community fencing has been a success in the Karaya landscape, and it's also cost effective and needs very minimum maintenance. So similar can be implemented in the district, uh, provided an assessment is done prior and post to that. And also, conflict concentrated training to the village business parties could be imparted. And this is the hanging solar fence which has been installed in the park boundaries of Mudumala Tiger Reserve and Manas National Park as well. And same has been initiated in the Karnataka National Park. So it would be very nice to see that how this would function. And it's very important to also emphasize on community awareness and educating the youth to inspire the minds to conserve the species. Apart from that, a combination of barriers such as the elephant tooth fences and the solar fence put together can be an effective way. And equal representation of the stakeholders is also very important. And it was noticed that most of the people in the area did not apply for compensation because it was a very cumbersome process, and that's why simplification of compensation application process is necessary. Apart from that, this will also bridge the communication gap between the police department and the local communities, and strengthening of harmless traditional mitigation measures and inclusion of early warning systems can also help in reduction of human and conflict in the district. And with this, I want to thank all these lovely people for helping with my dissertation, and without whom my study would not have been fulfilled. Thank you so much.
Question? This is a very nice presentation, nicely done, like nice topic also, and your my only suggestion is, since you are the 10 years of data, it would have been better to compare the same site before and after the diagram. So what you have done, you have compared a diagram versus a non-diagram, right? And there may be a few fields where you would have in that particular area. But if you compare the same site before and after diagram, that will give you a much right, that will give you power to say that yes, these diagrams are that
So actually, I could have done that, but the thing is that the solar panels in the Kaziranga National Park was built in or installed in 2007. So I did not have that pre-data, and because I considered only from 2010 to 2019. And for the solar trends, it was developed recently in 2024. I did not have that data.
Yeah, yeah. Yeah. So this is a very interesting presentation. You have a lot of information from the past and you collected some information yourself.
So, you're not clear.
Can you hear me now?
Yes, sir.
Yeah. So I said it's a very interesting presentation. You have a lot of information from the past as well as some data you collected yourself. The question is, you sampled a solar fence line of maybe 4 kilometers, is it?
Yes, sir.
Right?
Point.
Yeah. And then a trench line of 2 kilometers,
What is it?
a trench line of 2 kilometers.
Yes, sir. Yes.
Yeah. So do you think that length is enough for you to assess in terms of, whether that length justifies the variability along that landscape of elephant use and variable elephant use, variable density of elephants, variable people density, and economic status and whatever. So how do you sort of justify that part and of your sampling in terms of elephant conflict mitigation measures and its comparison, given all these variables in the particular landscape?
So, of course, it's not enough for elephants because it's a very smaller area to look, which I looked at. But when I tried to look at different physical barriers which were available in the district, I could find only these barriers apart from the Himalaya wall which was there. But I could not assess it due to lack of permission. So did not have any other ones.
Yeah, so I would suggest that maybe if you within the ambit of your data collection and within all of that, maybe you could try to segregate those aspects and see uh, whether the question is it effective or not is the main question, and the efficacy of a particular method depends on the situation at that particular instance. Okay, so it may change. It may be that solar fences may not be effective somewhere else, because the situation there is different. Okay,
So
and
that's why the physical barriers are often site specific.
yeah, so that's what you need to capture in that, and then you need to suggest appropriate measures at certain sites best suited for that site. Right, that is one aspect of it. And the socioeconomic data, whatever recommendations you gave, were they recommendations from the data that you looked into, or were they general recommendations?
Sir, those were recommendations I collected as generalized also and also that were suggested in guidelines which were provided by the ministry for reducing.
That's fine. I mean, they give general guidelines, but with respect to your study and with respect to your site, I think you need to be more specific in terms of what you studied, what you found, and what you recommend. These general guidelines everybody knows, so keep them as a general guideline. But the specific you need to, because this general, after doing so much of study, then you go back to the general guidelines and it downplays your own study, right? So I think you need to stick to what you found and what you think is best for the particular thing, right? Okay, thank you.
Thank you.
Ridhima, good effort. Uh, my question is, it's rather a comment. Uh, I suspect this conflict zone has populations of elephants coming from different parts of that region. There are some elephants coming from Nagaland. Yeah. Okay. So I'm saying that the conflict issue, the conflict scenario that you see there, is pertaining to elephant populations coming from different parts, from different populations from across that region. Some coming from Nagaland, some coming from Karbi Anglong Hills, and possibly from Meghalaya. So I, I think we needed to have a better understanding of what these populations are, from where they are coming. Okay. Otherwise, these comments, these recommendations, it's very generic. And also, all of these elephants, they are headed down to to Kaziranga. Okay, so which was their original routes? Much of that corridor is fragmented and broken. So all of these conflicts will be there. There will be any number of studies. These conflicts will be there. Everyone will come up with suggestions, but that's not that. So, as from a research point of view, if you could predict which are those areas where there is still a possibility of maybe restoring a corridor. Okay. I mean, I might be digressing quite from what you had originally planned, but I'm broadly saying from the conflict point of view. And this really requires some amount of research on elephant populations and their behavior, how they're using this area. Okay, thank you.
Proper question?
Hello, hello. Slide is blank. So I would like to start by coming to the by saying that humans have left no place on this earth untouched and have significantly altered the Earth's ecosystems. This has prompted the study of the human component in ecology and the spaces they occupy. This has given rise to the field of urban ecology, which is the study of the urban environments. As the population increases there in the urban areas, the built infrastructure also needs to increase. This prompts a change in the size, density, and heterogeneity of the city, and this can be simply said to be the urbanization. Now, as the cities grow, a serious concern is how they expand into the surrounding areas. Cities usually do not tend to follow a very concentric road and they grow very irregularly. Uh, the urban core gets saturated and the urban core gets saturated and there is exploitation of habitats at the urban core as well as suburban and the rural areas, which leads to exploitation, which is basically the exploitation of natural habitats. This leads to fragmentation and habitat degradation, and this simply can be said to be the urban sprawl. Moving from this, we come to the urban biodiversity. The urban landscape is not only occupied by humans but also by many other species, and this is the urban biodiversity. Many studies have looked into the urban biodiversity, its documentation, and suggested challenges
and strategies for its better management. Now, we will look at the urban landscape, and I have broadly classified the urban landscape into two major categories. First, being the urban green spaces, being the urban green spaces, and these urban green spaces include remnant patches of vegetation, tree belts along the roads, wastelands, institutional grounds, and many such areas mentioned in these green boxes. And then there is the urban grey spaces. These are mostly the built environment or the artificial environment. It includes roads, buildings, plazas, malls, and so basically built and artificial structures. With this, I come to my study, which is making the urban matrix matter, in which I have looked at the characteristics of the informal community of the urban matrix and related it to the urban green spaces of development. So, why would one want to study the urban matrix? Firstly, research has majorly focused on urban green spaces and how the impact of these urban and in the context of the matrix, how the matrix habitats influence the green spaces. They are not looked at specifically the biodiversity of the urban matrix, but how these matrix habitats will influence the urban green spaces. Uh, there is need to analyze the urban matrix, and this is very relevant because the matrix holds significant biodiversity, which is shown by many papers. Also, the urban green spaces and the urban matrix are interconnected. There is permeability between the urban green spaces and the urban matrix, which allows movement of many different species, and this has been recorded by many different studies. Also, the matrix biodiversity, as I mentioned earlier, is underrepresented. There has been very less research globally into this urban matrix, and in India, it has not received much attention, and there are very few studies. So with this, I come to the objective of my study, which was firstly to determine the feature of the informal community in the urban matrix of Dehradun. To answer this, I looked at the richness, abundance, density, and composition of the informal community in the matrix. Secondly, to examine the informal community in relation to the urban green spaces, and to answer this, I looked at the richness, density, and overlap in the species in the informal community for the urban green space and the urban matrix. So my study site was Dehradun City. Uh, I followed a previous study of done in similar landscape which focused on the urban green spaces. This was done by Kaushik et al. 2020. So in the map, the large green spaces which you can see are the urban green spaces, and the black dots around it represent my sampling points. The data of the urban green spaces was taken from Kaushik et al. Study. So my methodology was for the bird surveys. My sampling period was from March to April 2021. I placed variable radius point counts around the urban green spaces at 50 and 250 meters away from the urban green spaces. It was made sure that there was at least 200 meters distance between each point count station. It was followed by, at each point count there was 10 minutes of sampling, which included 3 minutes of acclimatization time and 7 minutes of data collection. For analyzing my patterns in the matrix, I first did habitat characterization around each point in a 100-meter radius. This was done by using Google Earth imagery and classifying the habitats or the land cover types into 4 5 major categories: built up, green cover, open areas, scrubland, and agriculture. This was for the landscape heterogeneity was calculated. The landscape heterogeneity around each point was again calculated in the 100-meter buffer using the patches of a Shannon diversity for the patches of the different habitat types. The major habitat types around each point were then introduced in a clustering algorithm, which clustered my sampling units based on the dominant habitat type present at that particular sampling point. Estimated species richness was calculated using the improved curve estimator by Chivatal. Density estimation was done in distance software for the sample tree. I used non-metric multidimensional scaling to look at the differences in the bird community composition of the matrix. Linear modeling approaches were used to quantify the impact of the habitat, quantify the impact of the land cover types on the bird community. For the comparison of the urban green spaces and the urban matrix, I plotted a simple Venn diagram to look at the difference, to look at the overlap between the communities of the urban matrix and urban green spaces, and simple box plots to look at the differential density and richness. So now come to my results. 3,775 individuals belonging to 81 species were recorded in the study area. The top five species were feral pigeon, rosebill parakeet, red-belted bulbul, common myna, and jungle babbler. These accounted for 41.67% abundance of the total abundance. The clustering analysis based on the habitat types resulted in five major clusters being formed based on a dominant habitat type. And this table shows the 109 sampling points divided, distributed into based on dominant habitat type according to, so they are clustered into their dominant habitat types. And this was used for further analysis for the habitat-wise classification of habitat-wise characteristics of the community. Species richness, the accumulation curve showed that build-up at the highest number of species, followed by scrubland, green cover, open area, and agriculture. The curve does not show saturation because the curve does not show saturation because the sampling was not enough to fully represent the species richness or different habitats in the matrix. And one of the reasons I could not do multiple replicates because of the pandemic restrictions. The estimated species richness showed that for all the habitat types except green cover, the observed and estimated species richness were almost similar. But in green cover, there was a bigger difference in the observed and estimated species richness, and this can be accounted to the fact that these plots of green cover showed more rare species, and these species were usually habitat specialists. They were not found in any of the other habitats, whereas across the habitats there were some common generalist species seen. Because of the presence of more number of rare species, the estimated species can be included. Abundance and dependence. So, in the four habitat categories agriculture, green cover, open area, and scrubland the community was almost similar in their evenness and abundance. But if you look at built up areas, it showed a highly uneven community, and also some species were dominating the community by greater margin. The density plot shows that green cover had the lowest density among the habitats, and this is counterintuitive to our thinking. But this green cover is of the urban matrix and not of a pristine forest. Here there was, as I explained before, there were a lot of rare species, and they were only seen once or twice, and the numbers were also pretty low. So this can account for the lower densities, but in the other habitats, more generally species are observed. Their abundance was also pretty high because of the presence of built-up areas, more human interactions and more human effects. The species composition ordination graph shows that the plots of green cover were clearly separated from the other habitats, and this is due to the presence, as I mentioned before, of these woodland raptors, hornbill, and woodpeckers, which were only found in the green cover plots. But in the other four habitat types, there were a lot of generally species like the feral pigeon, red-necked bulbul, and common myna seal. So this can account for this. Basically, is moving towards biotic homogenization in these plots in the other habitat types. Now coming to the habitat, coming to the borderless species richness, we were seeing that as the percentage of open area and scrubland increased, the species richness also showed a clear increase. But for as the percentage of built-up increased, we could see that there was a moderate level of peaking, and the richness decreased as the percentage of built-up area increased. This is in line with the intermediate disturbance hypothesis, which many studies have also tried to look at, and say that at moderate to medium levels of disturbance, the species richness of urban areas does show peaking because these areas show a greater habitat heterogeneity. There is more space available for them, and the disturbance is not so so much that many of these adapter species, urban adapter and avoider species, they can also come and coexist in these low to moderate disturbance areas. For density, it was seen that as the percentage of percentage of scrubland in the matrix increased, there was a clear increase. Landscape heterogeneity showed a very showed clear positive influence with the density. Percentage of built-up has shown in many studies that the density decreases as built-up increases, and for the percentage of green cover, as I said before, that there were habitat, as I said before, regarding the rare species and the density being, the numbers being too low, and the density also was low. Now we come to the comparison of the urban matrix and the green spaces. In this, the Venn diagram shows that there were 64 species which overlap between the urban green space and the urban matrix. 50 species were unique to the urban matrix, and these are your habitat specialists, or these are these urban avoiders which would not want to come into large amount of human disturbance or build-up, hence they were only found in these urban green spaces. The 17 species which are unique to the matrix can be accounted for by the fact that the sampling was carried out over different years, and also I might have seen some species which are rare, which may have come opportunistically in my sample. So the comparative species richness shows that the richness in the matrix is definitely higher than the richness in the the richness in the urban green space is definitely higher than the richness in the matrix. This is because there is more habitat heterogeneity and stability in the urban green spaces, where they can occupy more habitats inside there, and hence the species can also be higher. In terms of density, also it was a stable state where the urban matrix showed very low densities of birds, whereas the urban matrix, the urban green space clearly showed a higher density. There is supports higher density. The conclusions from my study: the urban matrix of Dehradun does show significant habitat diversity and a good proportion of green cover. There is substantially diverse avifaunal assemblage. Built-up areas are clearly shown to have a negative influence on the bird richness and density, and this has been corroborated by many studies. The landscape heterogeneity of Dehradun's matrix has a positive influence on bird density, and this is good because the urban matrix is not completely inhospitable. And you can see here that the bird species richness of the green spaces is greater than the richness in the urban matrix, but this does not mean that the urban matrix of Dehradun is inhospitable. It does have a substantial bird diversity, and this urban matrix needs more research attention in the future. I would like to thank these people, especially Nidhi, Shweta, Vidya, and everybody.
Questions?
It's a nice study which you have done first time in like in Dehradun Valley. My two questions I have. Do you think your 50 points for catching heterogeneity in the urban matrix is adequate? That's first question. Second thing, I have seen you know most of this is structural means the what plant species you have got, like especially Dehradun where you have lot of fig trees. Okay, so how have you taken any account of those species, the tree species in your green urban matrix? Because I have seen in the Matawala Park, which is in the heart of the Saranpur chop, and lot of bird species they come like hornbill and like that one. So I don't know whether how you try to capture those heterogeneity in the smaller spaces of those, and particularly the tree species which are conducive for bringing more diverse bird species.
So thank you for the question. And so I had a total of 109 points in the matrix. In 150, I had 109 points in the urban areas of Dehradun. And coming to the second point about the vegetational diversity which you mentioned, I had planned to look at the vegetational diversity, and it was part of one of my objectives submitted in the proposal, but it could not be fulfilled because I had kept the vegetation sampling towards the end of my, towards the end of my bird data collection. And because the pandemic restriction at that, I couldn't collect that, so I would drop the vegetation. But yes, studies have shown that vegetation is a very important aspect to look at when it comes to explaining the diversity, not only in the matrix but also in the green spaces. The vegetational heterogeneity does influence how which species occupy and how much numbers they can occupy. So yes, the vegetational diversity aspect can be looked at in the future. Sir, I hope I answered the question.
That's nice. So it would have been interesting if you have mentioned the area, like your study area in terms of like how many layers and how many people you have. And you can have made a relationship between the number of points in the area of the range.
Yes.
So, so you have a system of species that is common between these species and the matrix. Now, if somebody asks this particular question, yes, are these species because of the matrix or because of the green species? So, what's important?
So both are equally important, I would say, because you cannot have these 62 species colonizing these urban green spaces without the presence of a hospitable matrix. So if you were to consider an island biodiversity theory for urban areas, that would not work very well. Because if you say that these green spaces are like islands and the matrix is not suitable, then even not only for birds but for many other taxa, it would be not possible to find that. So once they are inside, there will be no more movement in and out, which actually adds to the species richness of both the matrix and the urban green spaces. So we looking at an island biodiversity view to saying that yes, 64 species, which is more important, both are equally important, and both need to be managed and conserved equally. Yes.
Okay, last question.
Yes, sir.
Because of this urban matrix, if we allow this urban matrix to expand, do you think the abundance of the common species will increase? And that abundance of the common species will have a competitive advantage for the species in urban green spaces, which will result in the decline of specific species or the rare species which are using those urban green spaces.
Yes, sir. You're right. You're very right in saying that cities are growing. There is so the matrix will grow, and as this matrix grows, as this keeps on sprawling outwards, there are going to be more and more green spaces formed. Which there are going to be more and more green spaces formed, which are as you can see WII, FI. These are all these green spaces formed because of the spread of Dehradun. And it is so these common species which you refer to in my study also the common species like feral pigeon, common minna, these kinds of urban exploiters. So they grow very well as the urbanization increases. So their numbers grow higher as the urbanization increases. There are urban adapters which work well with moderate levels of urbanization, and these avoiders, which you mentioned will go extinct. Yes, as the sprawl increases, as the number of these common species increases, these urban avoiders, these rare species that require particular habitat to survive will decrease, and the richness will decrease. So these few species like uh, common man of percussion are causing a comparative pressure also on these urban adapters and urban avoiders.
So is there a need when we look at the urban ecology? Is there a need to look at this as a different yardstick in comparing density, diversity, and comparing the presence of the layer in various
Yes,
species?
so people have looked at yield richnesses, so they're trying to classify them based on nesting yields, foraging yields, and also these urban yields, desert exploiters, and also looking at a finer scale will definitely help in teasing about these differences. Yes, I couldn't do it much because I had only one round or something, and the species also were I could not look into the yield richness that well and couldn't explain it that well. So I did look into it, but I couldn't explain it that well, hence I left it out.
Uh, Jason, my comment is relating to see these urban matrices that we are talking about spread across the landscape. from what we have shown in the map. Okay. Now, each I feel that in your overall summarization, the site-specific occurrence of certain species is lost, and it is getting generalized. For example, it is getting generalized. For example, if you take FRI campus, that particular patch, you are considering that as an urban matrix. You are considering that as an urban green space.
FRI is an urban green space, space outside as the urban green space.
Right. Now, if you look at FRI, the habitat connectivity with the hills, okay, that is going to influence what species is there in that area. Many of these are migrants which are moving down. That's going to be there in your data. A little beyond that, there would be certain species that will occur, okay, but there would be another site. See, what I'm basically trying to say is that if you take a site which is very far from FRI, say right in say Kantagar, many of those species would not occur. What you have there in the urban green space there in FRI, just taking some 100 meters or 200 meters away from that does not really explain to you the urban matrix.
I have taken points. So this is the FRI question. We have not taken FRI as in
Right.
this case, but these points, this is near Kandagar, this is near the core of the city, and these are towards the fringes. Yes, I have looked at that, and also while looking at the habitat coordinates, we did take distance from the impact forest patches, the nearest and the largest into consideration while running the linear models, but they did not come out as significant variable, significant in explaining the richness and density. We also looked at the distance from the urban green space and the size of urban green space. Both did not come as significant. So I accounted for the distance from the green, from the as you say, from the forest patches towards the Shivalik hills, from the Masuri side and from the Radhari side. We accounted that distance and it did not come significant in the analysis.
Okay, thank you.
Any further questions? Okay, then let's do a clarification.
Hello. A very good afternoon to all. As we all know, amphibians have very diverse life history strategies, and they are one of the curious organisms to look at. Right. The life cycle of a frog also makes very interesting observation for us. Now, they have very different life history strategies according to the areas or habitats where they occur. What about the cold and temperate regions? In perspective of an adult frog, some of us might have heard about some frogs are freeze tolerance as an adaptation, or they shift their breeding phenology to some specific season to adjust for the colder temperature situations. But what about the tadpoles, the non-reproductive life part of a frog? So the tadpoles also have a strategy, and this has been documented in several regions of the globe. And the tadpoles do this by a phenomenon called overwintering. So, what is overwintering? In overwintering, tadpoles actually reduce their development or like temporarily stop their development and and grow more and wait for the next season to metamorphose. So, if we are looking at different studies and observations on overwintering tadpoles across the globe, most of the studies are coming from North American region. And our observation from India is one of the first observation on overwintering tadpoles. And this is from a very nearest place from here, from Missouri. We have also identified some overwintering populations in Devil City also and Hanoi, which are like most prominent tourist places around here. Now, most of the cities have been coming from certain locations, as I told, and if we relate this with our observation from India, the region where we found the sovereignty tadpoles are also facing a lot of threats. And this is one of the major threats is basically the modification of stream habitats where they are occurring. So my study area was Missouri, and as we all know, Missouri is facing huge water crisis, especially in terms of drinking water. Now that makes pay to my sorry that pays to my dissertation topic, tadpoles with a trick, and I studied the overwintering ecology of these tadpoles in the Himalayas to Uttarakhand. And this study was supervised by Dr. Abhijit Das and Dr. Jay Johnson. So, before leaping into the complexities and final step details, let us define what is overwintering. So, overwintering can be defined as the delay in metamorphosis at low temperature conditions in high altitude areas and latitude areas where the average temperatures are lower and growing season are shorter. Now, we can simply define overwintering also as the spending of the winter season as in the larval stage and not metamorphosing as a crop. Now, there are a lot of studies spanning from 90s where they have looked at the effect of temperature, food availability, light intensity, and other intrinsic factors or the microhabitat features which are governing the overwintering behavior. And all of these factors have an effect on the morphology and development of the tadpole. Now, coming to my study area, which is the Dhobigat stream, which is flowing parallelly to the boundary of Missouri Wildlife Sanctuary. I have selected a 500-meter stretch of the stream, and I have identified the mix of habitat types, and I have selected 6 pools inside this stream. And four of the pools are natural pools, and other four are modified pools. I'll explain what actually is the difference between these natural and modified pools. But in this, the B1 and B2 pools, we exclusively studied the behavior of tadpoles, and the other pools we looked at all other habitat correlates. Now, the major species occurring or the overwintering tadpoles are supposed to be the tadpoles of Nanorana milica and Nanorana vicina occurring in this region, which are like mountain stream level frogs. Let me introduce you to the study area. This is the natural stream. You can see Missouri Wildlife Sanctuary and tadpoles. You can see mostly scraping on the boulders. These are the modified pools where they have check dams. And inside these pools, you can find tadpoles on the bottom layer, like not very active, mostly in the substrate area. But in the post-winter season, they become little active and they start scraping on the check dam walls. And when the water authority or the management people clean the area or renovate the check dams, these tadpoles faces mortality. Keeping these things, I kept my objective as first to assess the ecological correlates of density of orbital tadpoles between its natural and modified pools. The second objective was to understand the morphological diversity of these tadpoles, and the third one to evaluate the variation in habitat availability and use by the tadpoles. And I hypothesized that the microhabitat usage of orbiting tadpoles varies with the temperature gradient available in the natural and modified pools. And the second hypothesis was there are multiple annual larval species occurring in this area, which are having overwintering tadpoles, and they occur in lower development stages. So there a question comes, why lower development stages? Previous lab studies and observation from North America have shown that tadpoles in lower development stage tend to overwinter more compared to the higher development stages because they have very sensitive organs developing in their later development stage which are sensitive to cold temperatures. Now, answer this or to explore these objectives, I inventoried the habitat. So the selected stream was classified into niche habitat types: pools, run, cascade, and ripple. We can see tadpoles in pool, runs, and ripple areas, but mostly the tadpoles are seen in pool areas because they are not mostly preferring the low flow areas. So we selected the pools. I hope you remember the structure of the pools and the type of pools we have selected. Now each of these pools were inventoried by placing transit at transit placed apart 2 meter intervals, and at each of these transit, at each 1 meter interval, we have collected the depth, water velocity, and substrate type data for the availability quantification. And apart from that, we have also collected the cover item. We have also quantified the cover items, canopy cover percentage, and water quality parameters. Now, to estimate the tadpole density, we used a grid-based sampling. Randomly placed grids were used with 0.5 meter size, and the tadpoles were counted inside these grids. Both daytime and nighttime we repeated these surveys in nighttime using torches of 25 to 200 lumens. And some modified pools cannot be like properly surveyed because of the conditions. Like mostly in the natural pools, either the observer uh, walked along the boundary and counted the tadpoles. In cases where this was not possible, we used an inflatable boat to traverse through the pool and count the tadpoles. We also collected various habitat parameters in each of these pools. To answer the questions on morphometry, we collected tadpoles and processed in the lab to take the morphometric variables and we staged them and we assess the LBRF formula. And all the standard morphometric variables were measured. Apart from that, we collected live tadpoles and we photographed and post-processed to get the body length, tail length, total length, and maximum height of these tadpoles. Now for the behavioral part, we selected 2 pools and in each of these pools in a 24-hour interval, each 3-hour interval, I have walked along the boundary and counted the visible tadpoles. And we also recorded the activity state of each tadpole, whether they are stationary, moving, feeding, or surfacing, along with the air temperature, water temperature, and relative humidity data. And we converted this tadpole counts in each of the session as the percentage of maximum number of tadpoles recorded that particular day. Now the major analytical methods which was the generalized linear mixed effect modeling to understand the best predictors of tadpole density and circular statistics and activity overlap estimation for behavior analysis. And the putative species classification was based on the Lagrange two-prong formula extracted from the previous studies and literature. And we did a hierarchy cluster analysis and PCA to distinguish the tadpole groups based on the morphometric variables. Then to answer the last question, we quantified the frequency of observed, available and use of different habitat class types, and we generated habitat suitability curves and the Euler's index to understand the habitat availability and use. Before coming into the result, I will show what is the temperature variation in natural and modified pools. If we consider only the major pools, which are basically classified into modified and natural, there is no variation in water temperature between natural and modified pools, but there is an exceptional case where the daytime water temperature was high in modified pools during the post-lunar period. Now, if we consider only the behavioral pools in study, you can see the air temperature are almost similar in both the habitats. But when we consider the water temperature in these 2 pools, modified pool had a higher day and nighttime water temperature in winter, and in post-winter, the modified pools had higher night water temperature. Now, the first question was, do density and distribution of tadpoles vary with season and stream modification? So, yes, they do. And in the modified pools, if we are looking at the night density decrease, the night density is depicted in the blue color, and the night density decreased across months from January to April in the modified pools, but a slight increase actually we can see in the natural pools. And if we are looking at the daytime density which are depicted in the light green color, we can see that there is an increase across the month in both the pool types. Now, what are the best predictors of these temporal and spatial distribution patterns of the overwintering tadpoles? We selected after doing the GLM analysis, we selected the best model, and the best model showed that water velocity, equator depth, water temperature, modification, sorry, an interactive effect between modification and night time, canopy cover and cover item richness were negatively influencing the tadpole density. And an interaction between canopy cover and water temperature, also the night time, positively influenced the tadpole density. Even the modification was a part of the model, it alone didn't explain any variation in the tadpole density. Now, the next question was, are there any changes in tadpole behavior and emergence pattern between the modified and natural pools? Yes, there is. If you look at the winter activity pattern, both modified pools and natural pools had a similar activity pattern and the overlap was 90.8%. Now, it reduced in the post-winter period where the modified population had increased their daytime activity, but the natural pool population actually reduced their, sorry, not reduced, but concentrated their activity towards night time. And generally, the behavior, if we look at, most of the tadpoles were stationary, surfacing and moving tadpoles were very rare in the population. Some of the tadpoles started feeding in the late April. And the next objective, first question was, what is the taxonomic grid of forwarding tadpoles? So, based on the morphometry, we classify the tadpoles into two groups based on the UPGMA cluster analysis. And we found two tadpole groups based on the morphometry, but this needs to be confirmed based on the molecular information. And PC also showed a similar result. And the third unidentified group, which is happening in PCA, can be potentially a lower development stage of PCNA. Now, in objective to the other question was, are there any changes in morphology of tadpoles between the natural and modified pools? Yes, there is. This is a very interesting pattern where the modified pool population had higher relative tail length compared to the natural pools, which indirectly tells that the body length of the tadpoles in modified pools are reduced. And even though the pattern was clear, the variation was statistically significant in the post-winter. Now, but then the question comes, is this an artifact of the tadpole relative tail length? But it's not. We tested that and the tadpole relative tail length was not, was not significantly different across species. In objective 2, the next question was, are tadpoles of relatively lower development stage are only overweighted, like the Lyetta hypothesis? So, before coming to that, we found wide range of body size class in all the months, which means that there are multiple overwintering cohorts. It can be like 1 year, 2 year, and 3 year overwintering tadpoles. It's actually have been shown that there are tadpoles which can overwinter for 4 or more than years. So, which needs to be confirmed based on individual tagging and further monitoring. And the development stage based on the Gosner staging, we found tadpoles are also overwintering at the higher development stages, which is contradictory to the Lyetta hypothesis, which also has been shown from North America in very recent studies. Now, in objective 3, the question was whether the microhabitat use of forwarding tadpoles varies between different food types in response to the temperature gradient available. So, when we generated the habitat suitability curve, it was very clear that tadpoles were preferring refuge areas, which means 0 meter per second velocity, and a slight usage of 0.01 to 0.2 meter per second velocity class in the post-winter period only in natural pools. This can be because the tadpoles are more exploring in the natural pool habitats for maybe resource or other variables. And when we looked at the depth, it was clear that the tadpoles were mostly preferring a class of 30 to 60 centimeter depth. And when it comes to the cover items, we found similar patterns where the tadpole preferred submerged vegetation cover, except in the case of modified pools during winter. In this particular time, the tadpoles were using long undercuts in the modified pools, which is an observed factor also in the field because in modified pools the substrate is almost uniform and there are no cover items available. So, apart from litter, which they are not much using. A similar trend can be seen in substrate also, where tadpoles generally preferred sandy substrate in post-winter in both the pool types, but they differed in that during the winter time, where they preferred algae in modified pools and humus in natural pools. And in the objective 3, I had these two questions: whether the diet composition and diet width of tadpoles varies across seasons, and also does body size influence the microhabitat selection of tadpoles. I didn't answer these two questions. The first one was due to I have collected the tadpoles, but I couldn't process due to COVID restrictions and other logistical limitations. And the second population of tadpole emerges in different scale, like diurnal scale and seasonal scale. It also varies in natural and modified pools. Water temperature acted as a negative influencing variable, and this is also in accordance with the latest study by Bonilla et al. It says that temperate tadpoles are not Patagonia are adapted to low temperature and they are not under any thermal stress, considering the moderate climate change scenario. And this also underlines a very important thing. We have to consider temperature fluctuation in this kind of habitats, not like mean temperature and the variation in density. And an important factor which came as an interactive effect here is canopy cover. Actually, canopy cover can act as a resource gradient. It can have an effect on water temperature, dissolved oxygen, and periphyton growth in those continuous pools. Now, we also identified presence of wide range of size classes, which potentially says that there are multiple overwintering cohorts in the study area. Also, advanced development stage does overwinter. Why this and how they are doing is another interesting area to explore. The increased relative tail length in modified pools we observed can be an indication of the thermal and food stress they are facing in the modified pools, which recently was proven in a lab study by Merila et al. And a general observation and from the behavioral data, we concluded that they hide during daytime in some refugia. They come out in the night time, and so they come out in the night time. And this can be there. There are two observed examples for this. For example, salmonids do this particular behavior for feeding, and smoky general tadpoles does this for avoiding predators in the daytime. We exactly don't know what is the factor acting here, but that is also a potential question to look at later. Variation in substrate and cover item preference and avoidance, as we see in the last question. This can be an indication of their preferring some refugia from predation, temperature, or flow, light and food resource availability. Now, this also asks a question to us: if this is a part of their behavior thermoregulation, then the modified foods does not have much diverse or enriched cover items. So how does this affect? That is also an interesting question to look at. Now, these tadpoles can act as potential model to assess the influence of temperature fluctuation on the larval period, the adult breeding and survival, because they are adapted to the seasonally dynamic environment and modified habitats. Now we identified two tadpoles groups based on the LPR formula, but which needs to be confirmed using molecular identification. The major conservation implication of the study are the pool habitat of the stream should be maintained with a depth class of 30 to 60 centimeter depth in the yield season, which is winter. And since I have already discussed the Himalayan streams are subject to large scale hydrological alteration, this study gives a way to conserve these kind of tadpoles and the population occurring in these mountain habitats. And the habitat suitability curves we have generated in this study can be used in any of the hydrological models to estimate the ecological flow required for the Nanorana species in the Himalayan region. The major limitations of the study was incomplete winter season data, which needs to be expanded. The molecular identification needs to be done, and the research on breeding phenology should be strengthened. All the specimens we have collected, the tadpole specimen, debrider, periphyton, sediment, and all we have to process, which will throw light on morphology, periphyton, physiology, and neuroendocrinology of these tadpoles. Now, to establish actual overwintering pattern, we have to tag them and monitor, and further studies based on the effect sizes reported in this study and design issues can be utilized. So, I hope you enjoyed the journey with overwintering tadpoles, and I thank these amazing people with me, my supervisors, Anshita, Swati, Rishabh, Shadul, and Krishnendu, and all these amazing people behind all this open source software which made this dissertation possible, Wildlife Institute of India, Forest Department of Uttarakhand, and XVII. Thank you.
Thank you. Okay. Yes. You have questions? It's a very nice presentation and very nice study. Congratulations. So I have just a couple of questions. How you calculate the density of the acres? Which method you have?
I actually counted the number of tadpoles inside the cage.
Okay, so do you think you need to correct this protection probability considering the fact that a lot of tadpoles will be hiding their food?
So, but actually, it can act as a a proxy of number of visible tadpoles which are not hiding. So actually it shows the emergence patterns of the tadpoles as in terms of length.
So then you have two issues. You have tape codes which are hiding, so you have two types of issues. One is the available, one is the tape codes which are needless, not visible to you. They are these are the different population. And the population of the animal is visible to you. What is the population which is not visible to you? Then the visible populations, whether you are detecting everyone or how you will account for the population is a big issue.
So, like I have an assumption that at least in night time, if the tadpoles are coming out. If the tadpole is coming out in daytime, they are not in night, or if they are coming in night, they are not coming in the daytime. So at least one time they should be coming out from their region. That's what my assumption.
So that's your assumption.
Yes.
Please log into increase the database and then only if you have a 50 page course market and if you speak about it, then you are not going to be able to that's what that's the way it's like. Talk about the abundance variation, we very especially part of the population. Second question is the site variation.
Have higher taillight, which means lower positions. So lab studies have shown that this can occur when the tadpoles are facing thermal and full stress. There is only one study, which is a lab study. But there are field observations across the latitude, this stress is happening.
And then you will then
Okay.
more and does this variation in size
We have to look at it. That's what this is.
It was a very interesting presentation. Uh, Regarding the tail length, I also had a sort of a question. You said that one study has said that there is some sort of nutrient stress, right, which has caused, which probably leads to some body length or body, change in body morphology or body size. So, did you sort of look what possible nutrient stress could be there? Because in terms of how these artificial pools were they very segregated from the natural ones? How many of them were closer to the natural ones? Did you look at the spatial distribution of both these types of pools and then sort of segregate, let's say, those ones which are closer to natural conditions, okay, and farther from natural conditions? Maybe they could be similar in terms of, you know, uh, tail length if they were, let's say, closer to the natural pools, but if they were really isolated, sort of artificial ones, there you had more pronounced differences in body length.
Yeah, so the spatial variation can be looked at, but what we have already collected is the periphyton availability in these modified foods and natural foods, also the cover, sorry, the leaf litter and sediment sample. We definitely will look at, but currently we have to process the data. So I think some interesting result will be open there.
So you do have some microhabitat data which can account for this variability. Okay.